We'll start here with my slides. So as I mentioned, we're going to be doing a training using DNA Subway's blue line today. If you're not sure what that means, we'll talk about that again in just a little bit. But DNA Subway is our bioinformatics tool that's going to allow you to analyze your ant sequence data. But before we get into this, we really want to talk about what is DNA sequencing. So I'm going to spend a little bit of time explaining what DNA sequencing is and how DNA sequencing works, because what we will see is the raw DNA sequence data in DNA Subway. It'll be the very first thing that we see is the raw DNA sequence data. So what exactly is DNA sequencing? Well, it's a technique used to determine the order of the nucleotide bases, so adenines, thymines, guanines, cytosines, in a particular region of the DNA. So remember, in DNA barcoding, we're not doing whole genome sequencing. We're looking at one particular region of the DNA. So we're looking for this type of sequence data from one particular region. And again, for our ants, we're looking at the CO1 gene, cytochrome oxidase 1 gene from the mitochondria. I wanted to mention a little bit about why we're able to do sequencing at the level that we are and with students and with citizen scientists. And that has to do with the fact that the cost of sequencing in general has gone down, specifically for the cost of genome sequencing. So if you take a look at our chart here from the NIH, what you can see is the predicted cost of genome sequencing to our current time was predicted to be a little bit over $100,000. So maybe here around $400,000, $500,000 or so. But you can see that instead the actual cost of even whole genome sequencing is substantially less. This is about $1,000. You can sequence an entire genome of an organism. Why is this? Well, during this time from about 2003 to, from about 1990 to about 2003, the Human Genome Project was going on. And a lot of technological advances were made during that time, which carried over past the end of the Human Genome Project. So scientists continued to refine the technological advances that made the Human Genome Project sequencing possible. So what this meant was that there was a rapid increase in the technological advances that allowed us to much more cost effectively do whole genome sequencing, which in turn also brought down the price of the type of Sanger-based sequencing that we use for DNA barcoding. So now we can sequence a read anywhere, depending on the service you use, from anywhere between $3 and $8 a read. So relatively inexpensive, just dollars to be able to get DNA barcode sequencing accomplished. So to understand DNA sequencing, and again, if you have any questions along the way, please do feel free to ask. But to understand DNA sequencing, we have to understand a little bit about the structure of the nucleotides in DNA. Of course, the nucleotides are the building block of DNA. They're made up of a nitrogenous base. In this example here, we have a guanine, a sugar, and a phosphate group. And you can see here, this is how they're connected in a DNA chain. They're connected via the phosphates in the sugar phosphate backbone, and between the bases in the center here are hydrogen bonds, relatively weak hydrogen bonds that hold the double-stranded DNA together. What we're going to focus on with regard to the actual sequencing is the structure of the sugar. So I have here the structure of the sugar in an RNA molecule and in a DNA molecule. And you can see that actually the sugar is where in part RNA and DNA get its name. So this is the ribose sugar in an RNA. You can see that there are 5 carbons labeled here. This is the first carbon called the 1', 2' carbon, 3', 4', and 5'. So if you're not familiar with chemical structure, it's very common when you're representing like a sugar or a chemical like this, that you won't write the C for a carbon. You'll just see that the 4 bonds that it can make coming from a central connecting location, that's representative of a carbon. So we have 1, 2, 3, 4, and 5 sugars in a carbon. Off of the 2' carbon in RNA, we have an OH. This is called a hydroxyl group. In DNA, we only have a hydrogen. So this is actually in part where DNA gets its name. So RNA, ribonucleic acid, that comes from the ribose sugar, but the lack of the oxygen, which is what deoxy stands for here, that's the deoxyribose that comes from deoxyribonucleic acid for the name in DNA. which is interesting. But what I want to focus on here is actually the carbons themselves and the numbering of these carbons. And we're going to look at the 3' carbon here and the 5' carbon here. And you can see for both RNA and DNA, on the 3' carbon, we have this hydroxyl group, okay? This may seem somewhat familiar to you. So if these numbers sound familiar, you know, 5' and 3', that's because we assign 5' and 3' to the DNA when we're referring to its directionality. So you might see a sequence written as 5' to 3', or here's one talking about the 5' end versus the 3' end. So if you take a look here in the structure of a DNA molecule, you can see here again, we have a nucleotide in DNA. We have the, excuse me, we have the sugar, we have the phosphate, and we have the base. And if we look at the carbons, we have 1, 2, 3, 4. This is the 5th carbon here. So we're starting at the 5' end of the molecule at the top here. And that carbon, of course, is then attached to that phosphate group. On the bottom side of this particular structure, you have the 1, 2, 3, 4, 5 carbon. The carbon that's facing down is the 3' carbon. So this would be, this strand directionally would be referred to as 5' to 3' in this particular direction. Now, DNA is not a parallel structure, it is an anti-parallel structure. So that means that the other strand on the other side is flipped. So it's essentially upside down from this strand that we have on the left-hand side. So now instead of it being 5' to 3' this way, it's actually the opposite. If you look at the carbons, 1, 2, 3, 4, here's the 5' carbon here, whereas the top 1, 2, 3, now the 3' carbon is at the top instead. So that's referring to the directionality here. So now let's focus on these 3' hydroxyls that are available in a growing DNA chain. Now, to do a sequencing reaction, it's very, very similar to doing a PCR reaction, but we add in an additional component. So we will have nucleotides available to do a sequencing reaction because we need to build new DNA molecules. So we will have the deoxyribose sugars within the regular nucleotides, but we will also have something specialized called a dideoxynucleotide. And a dideoxynucleotide actually has been chemically modified so that the 3' carbon here, instead of it having a hydroxyl, will have a hydrogen. Why does that matter? Well, because that free 3' hydroxyl is what's needed to continue synthesis in a growing DNA chain. The lack of the hydroxyl, so the presence of the hydrogen here in the dideoxyribose that we're seeing here, will end the synthesis reaction, okay? So I was showing you what the nucleotides were looking like inside of the DNA structure. So free nucleotides, what are actually incorporated into the PCR reaction, or in this case, the PCR and the sequencing reaction, you actually have the base, the sugar, and then instead of a single phosphate, you actually have 3 phosphates. So this is called a deoxynucleotide triphosphate for if you're making DNA, or just referred to as DNTP. So that's what I'll refer to this as from here on out. And then, of course, the ones that have that chemically modified 3' hydroxyl, so that's just a hydrogen now, those are known as dideoxynucleotide triphosphates. Those are DDNTPs. So this is used in that Sanger-based sequencing method. This is also known as the chain terminating method because the incorporation of this, again, into a growing DNA molecule is going to terminate the reaction. So again, this is an incoming incoming DATP. So this is an ATP DNTP here that we're seeing. This incoming 5' triphosphate that we're seeing here will interact with the free hydroxyl that we're seeing on the nucleotide that's already been incorporated to the chain. So what does that do? Those extra phosphates, that actually catalyzes the reaction. That's the energy that produces the ester bonds that are formed here, and also releases water as a byproduct. So now we have synthesized another nucleotide in this incoming chain here. So I want to just demonstrate to you how this works. And I'm going to share with you an animation that we have developed. Okay, can everyone see my animation here? Okay, great. So to sequence a piece of DNA, you need the same components that you would need for a PCR reaction to synthesize new molecules. So you'll need your template DNA. In your PCR reaction, your template DNA is your purified genomic DNA. In a sequencing reaction, the difference is instead of it being your genomic DNA, it is now your PCR amplified DNA. So in our case for US ants, that will be the amplified cytochrome oxidase 1 DNA. That's our template for a sequencing reaction. We'll need a primer that's complementary to our template DNA. So again, this can be the same primer, which is the cytochrome oxidase 1 primer that we use. And again, this is designed to be complementary to our DNA sequence, and that will direct the polymerase to that specific location to make copies of that particular region. You will need an enzyme to catalyze the whole reaction. Of course, the enzyme for PCR is DNA polymerase. It's the same enzyme that's inside of our cells that catalyzes the synthesis of DNA. And it's the same enzyme that's used for a sequencing reaction as well. And the 4 nucleotides, A, T, C, and G here. So again, that's the sugar, the trinucleotides, the sugar, the triphosphates, and a base. So all of this is combined into a tube, in addition to those dideoxynucleotides, those DDNTPs. So remember, each of these are chemically modified so that if they get incorporated into a DNA chain, they stop the synthesis reaction. And additionally, for cycle sequencing, which is a modern variation of the Sanger sequencing originally designed by Fred Sanger and colleagues, what's nice about the cycle sequencing is each one of these dideoxynucleotides is also fluorescently labeled. So A has a green fluorescent tag in it, C's have blue, T's have red, and G's have yellow. Sometimes G is represented, you'll see as black, just because the yellow bars or the yellow fluorescence can be difficult to see. So you'll see that as a black peak instead of a yellow. So these also get added to our reaction tube here. So now what happens? Well, we simply perform a PCR reaction. So what do? We heat our DNA up to about 96 degrees Celsius. This will break those hydrogen bonds and separate the template DNA strands. We cool this slightly so that our primer sequence can find its complementary sequence and bind to the template DNA, as you can see here. Still hot enough now, though, so that the template DNA strands don't come back together, but the short primer strands can bind. Then our DNA polymerase will come in and will bind to the template DNA. And this will allow us to start incorporating the nucleotides. Now, what the polymerase will do is it will randomly grab nucleotides from this pool of DNTPs and DDNTPs to incorporate into our growing chain. Now, when I say random, it means it can't differentiate between a DNTP or a DDNTP, but of course, it still has to pull in a complementary nucleotide to the base that's in the DNA chain. So, okay, so it's going to start to bring in these nucleotides. So you can see it's bringing in a T here, a G, a T, and it's creating this strand here until it pulls in one of those dideoxynucleotides, in this case, a DDATP. Now it's going to try to continue this synthesis, but remember, this DDATP here is missing that 3' hydroxyl. So what happens if we now try to add another adenine at the next spot, which would be the normal nucleotide we would add in this particular chain? It can't, okay? So it comes in, there's no hydroxyl for the phosphate on the incoming nucleotide to interact with, and it ends the synthesis reaction. Polymerase falls off, and that's it for this molecule. Now, I want to remind you that when you do a PCR reaction from a single starting piece of DNA, after about 30 cycles of PCR, which is what you were doing with your particular components, if you were doing PCR, you'll notice it was about 30 or so cycles. So from a single starting piece of double-stranded DNA, you can end up with over 1 billion target strands of DNA. So over 1 billion copies of that cytochrome oxidase 1 region that we were trying to amplify, our DNA barcoding, from just a single starting piece of DNA. So think about it. We sent about 20 microliters of our sequencing reaction off to GeneWiz to do the sequencing. And there are billions upon billions of our cytochrome oxidase 1 DNA in those tubes. So what does that mean? Well, what that means is that during the synthesis reaction, what's happening is that one of those dideoxynucleotides is being incorporated pretty much at every single possible location along the length of our template DNA strand. And our template DNA for cytochrome oxidase 1 is about 700 or so base pairs in length. So this is going to happen where we're getting a terminating dideoxynucleotide being incorporated at every single position on different strands. And in fact, to the point where this is actually going to happen probably thousands of times at every single position because we have billions of copies of DNA. So what does this mean? We're going to take this entire sequencing reaction then, and we're going to run a gel on it. So same as you were doing with an agarose gel, we're going to run these samples on another gel. But this particular gel is called a polyacrylamide gel. It works the same in concept as an agarose gel, which means it uses a charge to mobilize the DNA through the gel, and it also separates the fragments of DNA out by size. But what's important about this particular type of gel is that it is very large gel, it is very thin, and it's very hot. So what does that allow us to do? It allows us to be able to distinguish between different fragments of DNA by a single nucleotide. So we're looking at single strands of DNA, and we can differentiate between something that's one nucleotide in length versus 2, versus 3, versus 4, versus 5, and so on. Okay, so it's got much better resolution than an agarose gel has. Okay, so we're running this gel. This gets placed inside of a DNA sequencer. And again, a charge is applied and our fragments are going to begin to separate out on our gel, again, by size. So smallest fragments will come off first, larger fragments will come off behind. And what we're going to see is this little laser is going to read those fragments as they go by. So what is it reading? It's reading the fluorescence of the dideoxynucleotide that was incorporated. And again, each of those fluorescent colors was representative of either an A, T, C, or G. And we're getting these peaks then, okay, in kind of this simulated image that corresponds with A, T, C, or G. And the height of the peak simply has to do with how much DNA was in the band as it passed by the fluorescent, or excuse me, the laser that was reading the fluorescence. So what this produces then is called an electropherogram. You may also hear it referred to as a chromatogram. Basically, an electropherogram is the same thing as a chromatogram. It's just when it's done by running the DNA through a gel. So what we're seeing now is each of these peaks, because they represent the nucleotide or a base, a color of the base, we're now seeing the DNA sequence coming out from this synthesis reaction. So overall, how would these fragments sort out if we were to look at how this would work in a gel, all of these different sized fragments? So if you see here, let's say these are all the fragments of our sequencing reaction. When we run a gel, again, they sort out by size. Each one of them ends with a fluorescent tag. So what we're seeing now is the very first fragment. Okay, so this is one nucleotide past the primer. This is two nucleotides past the primer, 3, 4, 5, and so on and so forth. And what that means is that if we read from bottom to top, that gives us our DNA sequence. In this case, T, A, C, T, C, G, G, and so on and so forth up. So that's how we get the sequence data, or that's how we get the data, by doing DNA sequence. And this electropherogram is the raw data that we're going to get back from GeneWiz. So I wanted to make sure that everyone understood exactly what it was that we're going to see when this data comes back. I'm going to stop sharing there for a moment. So any questions on the DNA sequencing thus far? Everybody feels pretty comfortable with that? Okay.
Wait, I have one quick question. So sorry.
Go ahead, Mary.
So I understood everything up to this point for the most part. The thing that I don't understand is, I know we're looking for like a unique sequence from each ant, and it sounds like we're all looking for the same enzyme. So how is that? How do we does each ant have a different form of the enzyme? Is that what we're
So the enzyme will be the same. So DNA polymerase is the same for everyone. What we're looking for is the variation in the DNA sequence region, so that DNA barcode region. So one ant species compared to another ant species in that barcode region should have a different order of their A's, T's, C's, and G's. So that's what we're looking for. And we're going to take that and compare it to a database that contains thousands or even millions of these different sequences to see if we can get a match. And that's what I'm going to take you through when we go through DNA Subway.
Okay, thank you.
Sure thing. All right, so let me see here. Let me go ahead and share my screen again. So as I mentioned, GENWIZ here will send our PCR amplicon to GENWIZ, and GENWIZ provides that sequencing data back within about 48 hours. So it's a pretty rapid turnover time for us to be able to get the sequence data back. And then it's going to get uploaded to our bioinformatics website called DNA Subway. So what is DNA Subway? Well, it's an online bioinformatics workspace that was developed by the DNA Learning Center as part of this group called Cyverse. Also, it was formerly known as the iPlant Collaborative. And basically, this is an organization that provides cyber infrastructure to scientists and educators who are doing data analysis. And this was originally developed, DNA Subway was originally developed with funding from the National Science Foundation, NSF. So what does DNA Subway do? It takes all of these different websites that we would normally use to have to analyze sequence data, and it puts it in through an easy workflow so that we can in an easy manner, be able to analyze our sequence data almost in a step-by-step manner. So DNA Subway, of course, we call this kind of riding DNA Subway or riding the train, where we'll have different stops at different points, depending upon the DNA Subway line that we use, where we have to do some sort of task. And we will be focusing today on DNA Subway's blue line. But there, of course, are different lines of DNA Subway. You are always welcome to test out any of these other lines if you have an interest in the type of process that they offer. And so the red line will allow you to predict and annotate genes in up to 100,000 base pairs of DNA. The yellow line actually lets you prospect entire plant genomes for specific genes. The green line allows you to input RNA sequence data to measure differences in transcriptomes. Our newest line is actually the purple line here. That's our metabarcoding line. This allows you to put NGS metabarcoding data and metadata to measure and compare species in. So that's a really cool one if you have really large data sets. But we will again be focusing on the blue line. So this is where it will allow us to input DNA sequences to build phylogenetic trees, analyze DNA barcodes, and prepare DNA sequences for data submission. So if you would like to follow along, I'm going to give us some information in just a little bit on some example sequence data that everyone can use. But you can go ahead to the website dnasubway.cyverse.org. And Allison, I'll ask you to put that in the chat if you wouldn't mind. So dnasubway.cyverse.org, and you can follow along the sequence data that I'm going to be processing with you now. So the tracking numbers that we're going to use, so I should say the tracking, how you're going to get your data back from DNA Subway is through a tracking number. So I have two examples of tracking numbers here that we're going to use in today's tutorial. So you will get a notification once your tracking number has been returned from DNA Subway. I will provide you with this information, and you'll be able to then find your sequence data for all of your samples that successfully PCR amplified and successfully sequenced under this tracking number. And I'll show you how to acquire that in just a minute. But if you would like to follow along, go ahead and copy these numbers down. In fact, I'll go ahead and put these in the chat for you just to make it a little bit easier. Let's see here. So that's the first tracking number. And that's the second. So you can go ahead and copy those. We'll use those in just a moment. And I'm going to stop sharing and open up the DNA Subway web page. Okay, can everyone see that here? DNA Subway. Yes, okay. All right, so this is the home page of DNA Subway. You'll notice in the upper left-hand side here, you have the option to log in or to enter as a guest. For today, and you're welcome to follow along today by just entering as a guest, but for your US Ants project, please, please, please do register. So you'll just click on the register button here. This will take you to the Cyverse portal, which will allow you to then create an account and register with DNA Subway. If you enter as a guest, you can use all of the same tools that you would if you were registered, but the problem is it will not save your data. So I think it saves it for maybe up to 24 hours, but the problem is even if you keep that window open, it tends to log you out and then you won't be able to log back in. So we certainly want you to be able to go back and re-access your data that you've taken some time to analyze. So make sure that you register as opposed to just entering as a guest here, so that you can actually go back and re-look at your samples. But for now, I'm just going to enter as a guest. The other thing I want to point out is at the bottom here, there's some background information about DNA. There's DNA subway training. There's also a manual. So if you click on the manual, it will take you to a guide where you can actually click on a walkthrough of the DNA Subway's blue line. So if you want additional assistance in using DNA Subway, this is a great tool that will take you step by step through how to analyze data. And I believe we give you an example mosquito data set here for you to be able to walk through. Okay. All right, so let's go back to DNA Subway. Whoops, I think I closed out on my screen. Let me open a new one. There we go. Okay, so I'm just going to enter as a guest, and I was playing around with this earlier. So to create a new project, I'm going to just click on the blue box here that says Determine Sequence Relationships. So this will open up a new project under My Projects. And then once you have a bunch of projects or you've started analyzing your data, if you click on the My Projects tab, you'll see a list of projects here that you've started working on. So again, if you register, all of your projects will be saved under the My Projects. So click on the Determine Sequence Relationships box. And the very first thing that you need to do is to select your project type. So we have the option to select if you want to do specifically phylogenetics, especially with human DNA. We have that option here on the left. But of course, we're doing barcoding, and we're using the cytochrome oxidase 1 gene that we're using for the US ants. So you want to make sure that you select this. This is important for a few reasons. One, it will affect the example data sets that you can use if you want to walk through DNA Subway using different data. It will select ones based on whether or not you've selected RBCL or CO1. And then later, we can actually pull reference data into the project that we've created. So if you select RBCL, which is the plant DNA barcoding region that we generally analyze, it's going to give you plant data to compare to your ants later, which isn't going to make for, a very good phylogenetic tree. So make sure you select the correct primer up front here, which is CO1. The next thing it's going to ask you is to select a sequence source, and it will not let you select a sequence source until you've selected the type of primer that you're using. There are a number of different things that you can do to upload your data. You can actually upload or copy and paste data in what's called FASTA format, your sequences in FASTA format. I will show you what FASTA format looks like later. Essentially, it's just the raw sequence data. It has a caret, it has a title for the DNA sequence, a hard return, and the sequence data. So it sounds scarier than it actually is. You can, again, like I said, you can select a set of sample sequences. So if you want to practice, you're welcome to do that. But here we're going to actually import trace files from the DNA LC. That option is the sequence data that GENEWIZ has provided. So go ahead and click on that bubble. And what you'll see is the most recent number of cytochrome oxidase 1 related samples that have come through. Now, sometimes for some reason, it doesn't pick up the most recent CO1 data. I think it has to do with the example data set that the software uses to search for the sequences. So you can always just go in the upper right-hand corner here where it's saying showing orders containing CO1 samples and click show all. So if your sample didn't show up immediately, that should show up all of the most recent, whether they're CO1, RBCL, or other. And you can then click on your tracking numbers here. Alternatively, you can simply use the search bar here to enter your tracking data. And that's what I'm going to do here. So I have two tracking data numbers that I want to use to bring in data. The first is that 30-324-526-303 and click search. So that'll bring up my first tracking number, and I want to pull a couple of samples from this example to show you. One thing to remember, or one thing to be aware of, if you accidentally include a space before, so before here or after your tracking number, this will actually consider that a character. And if I hit search, nothing will come up. So if I give you a tracking number and you type it in and it's not working, go back and make sure that you don't accidentally have a space somewhere, and then your number will show up here. So that's probably the number one thing that I see people have problems with if they can't get access to their data. So go ahead and click on the tracking number. Okay, and what I'm going to do is I'm just going to select a couple of samples here. So I want PWH002 and PWH009. Now you'll notice for every single sample here, 1, 2, 3, 4, 5, so on and so forth, there's 2 sequencing results that show up. This is because, of course, DNA is double-stranded and we sequence both reads, both the forward strand and the reverse strand. So when you're working with your sequence data, make sure you bring both reads in, because that's going to allow us to create what's called a consensus sequence by comparing the forward read to the reverse read. So make sure you bring in both. For your sequences, I imagine for the most part, you'll just click the select all button, and that can bring in all of your data. But for me, I don't want to work with all of that. This is a lot of data for just an example. So I want to bring in just 2 and 9 here. Then scroll down to the bottom and you'll click add selected files. You'll see this green bar will start to scroll. One thing important to remember, I do not recommend using DNA Subway on Internet Explorer. It seems to work fine on Safari, on Mozilla Firefox, on Google Chrome. But what I found with Internet Explorer is when I import sequence data, what I'll find that green bar will actually get about halfway through and then it will freeze. So for whatever reason, this doesn't work particularly well on Internet Explorer, but other browsers, it seems to be no problem. Okay, so I've imported those 4 files, and you'll see that here it says successfully transferred 4 files. I'm going to go ahead and import more files from the other tracking number that I put into the chat, and that was 30-437-095-734. Search. And from here, I want to bring in sample RKH002, both of those, and add those files. Okay, so now I have all of the sequences that I want to use for this particular project. The other thing that you'll be required to do is to enter a title for your project. So we'll do US ants training. And then, of course, you're welcome also to write a description. Hopefully more specific than what I have here, but this can help you, especially if you have to go back to your data at some point. This will describe the type of data that you're working with. And then click continue once you're done. Now, this will bring you into your actual project with the sequences that you've chosen to work with. If for some reason you need to change the project type that you're working on, you can simply do that by clicking edit down here. So that'll allow you to change the description, the title, move it from a private project to a public project if you want other people to be able to view your project, so on and so forth. Just click save if you want to do that then. But here we have our stops for our DNA Subway Blue Line. So there are 3 stops: assembling sequences, adding sequences, or analyzing sequences. My recommendation is simply to just go in order. Start with the first stop here, and you're just going to use the buttons in order. Then you'll move on to the next stop and use the buttons in order from there. So it looks a little bit daunting at first, but it's actually quite user-friendly, especially if you just go in the sequence that we have designed DNA Subway to flow. So what is the first thing that we're going to look at? It's sequence viewer. So if you click on the sequence viewer button, and in fact, before I do that, I just want to point out this key down here at the bottom. So if you have the very first one you see here, it's a V inside of the dark green bubble here. That means there's something to view. So if you click on that button, there is something under that button that we'll be able to look at, that you'll be able to view. If there's an R here in the white bubble, that means when you click this, something will run. So some sort of process has to occur, something will run in the background. And usually when it runs, it will then turn into a V, so then you can view whatever was completed once you click that button there. While it is running, it'll flash. It'll be like this yellow button with the red R inside. Blocked here means that you need to complete a task before you can access any of those buttons. So consensus builder here, and phylip NJ and phylip ML here. Something has to happen before you can access those buttons, and I'll show you what that is in just a bit. Last thing here is error. I've rarely gotten an error, and usually if I got an error, it's because DNA Subway crashed. It's usually not because you did anything, maybe just the server crashed or I overloaded or something. So error is rare, but if you get an error, my recommendation is to just refresh the page. Okay, so let's go ahead into sequence viewer. So if I click on this button, what you'll see here now are all of the sequences that I brought in. So the forward and the reverse sequences for each of my 3 samples. You'll notice there's an extra R here at the end for sample number 2. That extra R at the back at the end just means repeat. So this is actually a sample that GeneWiz repeated the DNA sequencing on because the sequence quality was low initially. The other thing that you'll see here is a little box next to it. Most of them have what looks like little mountains in it, but those of course are the pictorial representation of the electropherogram. It's also known as a trace file. So if you click on that box, you will see exactly that trace file that I showed you in the DNA sequencing, which is here. You'll see these peaks represented, the colored peaks that represent the A, T, C, and G nucleotides. So what do we see here? Well, at the very beginning, we see the ends, and ends are obviously not A, T, C, or G. So N basically means that the software was unable to identify what nucleotide should be at that position in the gel, okay? You'll see that it's very common for us to have low quality sequence dating. So a bunch of those ends at the beginning, and you can see that there's no really distinct defined peaks here. As we scroll through, those peaks get taller and more distinct, defined. And then we go to the other end here and we again see that data start to become low quality again. So this is common for DNA sequencing results. Why is that? Well, at the very beginning, so remember I said when we ran that polyacrylamide gel, it separates the DNA strands based on a single nucleotide. Well, those first few nucleotides that come off of our agarose gel, or first few, I should say, DNA strands that come off the gel, you know, something that might be just a few base pairs in length, so the primer plus a few base pairs, and then, you know, one additional 2, 3, 4. Those are still really, really, really close in size, even for a polyacrylamide gel. So instead of getting distinct bands for really, really small fragments of DNA, they'll still kind of smear together. So that's why we'll get low quality here at the beginning until you get to about maybe usually 80 to 100 base pairs where it will start to clear up. And we'll get the much more distinct fragments, and you can see those bands in the gel much more clearly. On the other side, you're then starting to run out of the nucleotides, the ddNTPs, depending upon the length of the sequence that your DNA, your polymerase is trying to sequence. You might start getting misincorporations or mutations in the actual sequencing. That depends on the fidelity of the polymerase you're using, and so on. So that can lead to lower quality sequence at the end. So this is not uncommon. The other thing I want to point out here is what you're seeing here, these kind of purple bars here above this blue, above or below, I should say, this blue line. So this is called the Phred score, P-H-R-E-D. And if you see this little question mark here, it says, what is this line? If you click on it, it tells you exactly that. The Phred score is this measure of quality control. And if you were to click more here, it will take you to, I believe it's a Wikipedia page talking a little bit more about the Phred score. So what is the software measuring with this Phred score? Well, basically for every nucleotide, it's assigning a score between 10 and 50 in increments of 10. So it can be assigned 10, 20, 30, 40, or 50. If it's assigned a score of 10, what it's telling you is that 1 out of every 10 nucleotides is a potential misincorporation, or it's potentially incorrect, okay? If it's a score of 20, this is an exponential increase. So instead of it being 10, 1 out of every 100 nucleotides could be call wrong, base call wrong. If it's assigned a score of 30, then 1 out of every 1,000 nucleotides has a potential for being miscalled. Now 20 is our cutoff, so that's the threshold here. So anything above 20, so 30, 40, 50 is considered by the software relatively good, relatively high quality. So you can see here these lower peaks here, these are about probably 10s. Why are those considered 10s? Well, if we actually look at the electropherogram data, you can see there are overlapping peaks here. So that's low quality. So a lot of times these overlapping peaks here will be representative. We'll have lower scores here, whereas the nice clean peaks here, they are all given a score of 50. Okay, so that's very important. What we want you to do when you first look at your data is to scroll through each one of your sequences and make sure that it looks like you're getting a good quality score. Because if you have low quality sequence data, then our trust in the fact that that's the actual sequence data for your organism is much, much lower. So we want to make sure we have high quality. Now we do have something in place to help identify if you have low quality sequence data, which is this here. Instead of having the image, which is the peaks of the electropherogram in the box, we instead have this exclamation point inside of the triangle. So this is indicative of low quality. So what would that look like? Well, if I look, you can see here Okay, yeah, this really doesn't look good. You can see for the most part, the majority of this sequence does not kind of pass that quality control. And if we artificially enhance the y-axis here to look at the peaks, you can see the reason why is there's simply so much background here. There's so much overlapping peak that we can't get any good sequence from it. Now, what's interesting is that you can get low quality for both of your samples, so your forward and your reverse. And if that happens, why do we get a low quality score? Could be for a couple reasons. Could be that you accidentally contaminated your DNA with one of your other samples. So what it's doing is it's sequencing both of those samples at the same time, and it's getting overlapping sequencing data. That's low quality. Could mean that the amplification of your sequence when you did the PCR wasn't great. So you got maybe not a bright band, but maybe just kind of a modest or a low brightness in your band on your PCR amplification. So there's not a lot of DNA here. And if there's not a lot of DNA, then it can't confirm the quality of your peaks either. So those are the two most common reasons you'll get low quality sequence. The other could simply be an error during the sequencing reaction, which is why sometimes we'll get a low quality read for one of them and a higher quality read for the other. If that happens, and that's why specifically I brought in sample 9 here, I want to show you how you can still analyze the other sequence as long as the other sequence is high quality. And you can see as I scroll through that for the most part, yeah, this sequence is still high quality. You'll notice the peaks are a lot shorter. I can artificially enhance them. You can still see, even though it's got less of a signal, they're still clean. So the software still confirms that it's a high quality sequence here. So we can actually use the one read as opposed to both as needed. Okay, so that's sequence viewer. And again, you'll go through and you'll look at this for every single one of your sequences just to confirm that we have high quality sequence data for all of our samples that we're working with. Okay, so you'll close out of that window. The next is sequence trimmer. Again, we're not very, overly clever when we come up with these things. We like to tell it as it is. So what are we going to do with sequence trimmer? We're simply going to trim your sequences. So the software will actually automatically trim through your sequences itself. So if I click sequence trimmer, you saw it flash. Open this window again. It'll tell me my sequences have been trimmed. So what did it trim off? Okay, it trimmed off that very, very beginning of the low quality sequence data. It'll trim up until it starts to think the data is getting a high enough quality where it passes that threshold. We can trim this further. I'll show you how to do that in just a bit. But it trimmed off that low quality sequence at the beginning and the end for each of your sequences here. So you can see that. So that's just a nice feature to help reduce the amount of trimming that we need to do. Okay, that's all we have to do with sequence trimmer. Now, pair builder, what this is going to do is align your forward and your reverse sequences for all of your samples. So I'm going to open pair builder. We have this nice feature here that says try auto pairing. And what this will do is it will actually try to determine which pairs should be made automatically. So it did a good job here. It paired the forward and the reverse for each of my samples 2, 9, and RKH002 here. So if all that works well, you can go ahead and click save. I do want to show you quickly though how to manually pair your sequences. Why might you need to do this? Well, I'd say 99% of the time that I've used DNA Subway, the try auto pairing has worked perfectly. But if I have samples that are really, really close in sequence, or more specific, more commonly, I would say, if I have a sequence where I have maybe a low quality initial reverse sequence, say here, and then a repeated sequence that GENEWIZ did for me, it won't necessarily know which one to pair to. So you'll have to specifically tell it which sequence to pair. So how do you do that? You just have to make sure you designate the correct forward and reverse. And again, that will be designated here by the F or the R at the end. So if this is my forward for 002 here, click on the F for the reverse. That'll switch that to a reverse sequence. So it knows that that's a reverse. And you'll click both the boxes. Following that, it will ask me if I'd like to pair those sequences. Yes, I would. And now that's all set. Okay, so then I could do the same thing for my other 2 sequences. But I'm just going to go back and do the auto pairing and save everything. Okay, so now we're moving on to Consensus Builder. And I want to just share with you quickly what Consensus Builder is. Or what Consensus Builder, I should say, is trying to do. Okay, so when we build a consensus sequence, basically what we're trying to say is the forward sequence agrees with the reverse sequence, and we're coming up with a consensus between the 2 sequences. So what is it looking for when we do the consensus? Well, here's a couple of examples. For the forward sequences, we have, again, it's written 5' to 3', so we have the forward sequence. The reverse sequence is written 3' to 5' initially. So what we need to do is we need to create the complement of that reverse sequence. So if the first here, if this is a T, of course, the complement to the T would be an A. Okay, this is an A, complement to an A would be a T, so on and so forth. If the complement is designed correctly, then, or I should say, if the complement is exactly paired perfectly with the forward, then what we should get when the complement is formed is the exact same sequence as the forward. So essentially, we're just comparing the forward sequence to essentially itself, okay? If the reverse sequence is an exact complement to the forward. So what happens when it's not? And this will happen every so often. So here I have an example where the majority of the reverse sequence does complement the forward, okay? But here at this position where I have a G in the forward sequence, in the reverse, in fact, I get an A. Why is that? Usually has to do with something in the sequencing reaction. Maybe we got an overlap in peaks, could be there was just low quality sequence data here, any number of reasons why we get a mismatch in what we call here. So what does that mean? Well, when we then create the complement of this reverse strand, instead of this being the G that you would see in the forward strand, it's now just the complement of the A that was in the reverse strand, which is a T, okay? So what the software has to do is then make a determination between that position and that position. So I have that aligned here, the forward sequence and the complement, and make a determination in the consensus as to whether it feels it was a G at that position or a T at that position. And what this will look like in DNA Subway is it will be highlighted in yellow. So if you have a mismatch between your forward sequence and your reverse sequence, it'll let you know. It'll highlight it here in yellow. So again, we see the G here on the forward and the T in the complement. What we can do is actually look at that position in the trace file, in the electropherogram, to see why it was a G in one spot in the forward and a T in the reverse. So if I look here, the G in the forward, we can see, okay, that's a nice clean black peak that we're seeing here. But in the reverse, we see that same black peak, but in the background, it's overlapping with a red peak. So it had a hard time deciding whether or not it should be that G, excuse me, or that T. So it made the decision, this example, to put a T here, but you'll notice it's light gray. Light gray means that it didn't pass that Phred score, it didn't meet that threshold for being a high quality sequence because it's overlapped here. So it made a prediction, it thought it was a T, but it wasn't sure. So that's why it's light gray here. But the reason it chose the G is because that was a clean peak that passed that threshold for a high quality sequence. So you may have this when you look at your sequences. So I'm going to go back to DNA Subway now. Okay. So now we're going to do consensus builder. So we're going to do exactly what I just explained. It's going to align the forward and make the complement of the reverse and determine the consensus for the two sequences that we get back. So that's what happened when it was flashing. I'm going to now open consensus builder, and it's going to show me my 3 paired sequences here. And now I can scroll through and I can look at the alignment of the forward and the complement of the reverse throughout the entire sequence. Now, one thing I want to point out, doing the sequencing both reads actually gives us a longer DNA sequence to work with because you get more data on the reverse at the beginning and more data on the forward here at the end. So you can actually end up with a longer DNA sequence than if you were to just do sequence 1 forward read or 1 reverse read. The only problem is that you don't have something to compare it to. So you just kind of have to take it at its word here if the that in fact the consensus is correct based on just a single read. But we actually have the option to remove any sequence we're unfamiliar with as well. I'll show you that in just a moment. But you can see there are a few spots here where we have these yellow mismatches here, 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 and here. And if I click on this, so here we have the consensus sequence, you can click directly on the consensus sequence and it will open the trace file. So here it shows a C at this position in the consensus because it had a high quality C in the forward strand, and that's because we have this nice clean peak here, blue peak. Whereas on the reverse, it shows a T because there was this red peak here, but that was an overlapping with that blue peak in the background. So again, that's why we're seeing that here. And any other low quality sequence, it's where here we have an A on the forward and an A on the reverse, but you can see the A on the reverse is that light gray again. That's because we had that nice clean peak here. We also have that peak in the reverse, but it's again being overlapped with this kind of blue peak here. Now, I would say if you have a high quality nucleotide of one strand and a low quality nucleotide on the other, but it's the same nucleotide, then you can still be pretty confident in the consensus sequence here. So when I go through and I hand trim these sequences further, I will usually leave these sequences in. I'll just remove the mismatches. Now, when you trim your sequence data, you can only trim in from the ends. This makes sense because if you were to try to remove a chunk of DNA from the middle of the sequence, you're actually changing the DNA sequence, and we can't do that. We can only trim in from either side of the DNA sequence. So if you end up with one of these mismatches in the middle of your DNA sequence, I don't recommend that you trim all the way through to the middle and get rid of that sequence because you're going to be left with a very short DNA barcode region that you're going to use for comparison to this GenBank to identify your species. Why is it bad to have a short DNA sequence? Well, for example, if you were to have a DNA sequence that's just ATTA, right? And we took that DNA sequence ATTA and we compared it to a database of thousands or millions of DNA sequences, how many sequences do you think would come back with the result of ATTA in it? Okay, plenty, okay? And the first result that comes back, the probability of that in fact being your sequence is really, really low. So the larger your sequence, the higher the probability that the results that are given back to you are in fact a close match to your species or an identical match to your DNA sequence of your species. So our absolute minimum for being able to analyze sequence data on DNA subway is at 300 base pairs. Although we generally don't recommend going below 400 base pairs because then you start to get a lot of results back that a lot of matches to your particular sample because you're looking at a much smaller region and you're missing the regions that might have those polymorphisms or changes in DNA that could be distinctive between your species and other species. So at the very minimum 300, certainly recommend 400, but as long as you possibly can is better for doing this identification. Okay, so I'm going to go back to the beginning and I'm just going to hand trim your sequence. Now this is a little bit subjective. So where you want to trim is up to you. I'm more of a conservative trimmer. So I'm going to click on the trim consensus option here. You'll notice that the consensus sequence became white and I now have this gray bar that I can use to click through on my sequence. So I'm going to scroll over and I'm going to trim through both of my mismatches here and trim out all of that data that I don't have something to compare to. So I'm going to trim here and you'll see that a red line comes through and that'll indicate to me all of the sequence data that'll get removed. Then I'm going to scroll to the other side. Same here, I'm going to trim in and it's going to trim in and remove all of that data. Now again, there's a few of these gray nucleotides here on the reverse, but because they're the same as the ones on the forward, I'm going to go ahead and leave those in. You'll see that it'll tell me the total length of my nucleotide that I was originally working with. It was 719 base pairs, not my nucleotide, my DNA sequence, I should say, was 719 base pairs. And I'm removing 84 base pairs from the left and 87 from the right. This will still give me plenty more than that 400 base pair cutoff that we use for to make sure we have high enough quality DNA. So I'll go ahead and click the trim. That'll get rid of those missing sequences on either side. If I want to see what the final trim length was, I can just click trim consensus again, and it tells me now that I'm still left with a sequence that's 548 base pairs in size. So that's good. Okay, I'm just going to quickly do the same for my other sequences. Oh, actually, one thing I want to mention. So for sequence number 9 here, I'm actually going to go back and I'm going to unpair this because remember, The forward sequence for 9 was low quality, but the reverse was high quality. So I don't actually want to pair those two sequences together because it's not going to give me correct data. I only want to work with the one read. So again, this is another reason why I want you to know how to be able to do this manually. So I'm going to unpair those and just click save. Now, what that means is under Consensus Builder, I do not have the option of trimming just a single read. So you'll have to blast the entire read and just be aware that there may be a few mismatches on either end. Now, you can manually download the sequence and kind of cut out any ends that are at the beginning or the end if you would like, but usually just blasting the one read should give you a close enough match. So I wouldn't worry so much about it. Okay, so I'm going to quick trim my other sequence here. There's an N, you can see here, there's a light gray N in the forward sequence here. I do like to trim through the ends if they're at the beginning or the end, just because again, it's not telling me what base was there, so I have nothing to compare to. So I'm gonna trim through there, I'm gonna trim through here, and I still have 517, so we're good. Excuse me. Okay, so we are done with assembling our sequence data. We viewed it, make sure it was high quality, auto trimmed it, paired it, build the consensus, and further trimmed the consensus, okay? So any questions so far about how we've processed our sequences? Is everybody feeling pretty comfortable with that? Yeah, it's pretty straightforward. Okay, so now we're gonna move on to the add sequence stop here. The first and probably most important button we have here on DNA Subway is this BLAST button. So BLAST stands for Basic Local Alignment Search Tool. So what is BLAST going to do? It's going to take our sequences that we are, and we call those the query sequences, our sequence, and we're going to compare those sequences to a database that has thousands or hundreds of thousands or even millions of sequences available in it, which have been taxonomically assigned to some species or another, okay? The database that we are going to compare our sequences to is called GenBank. And GenBank is really the authoritative database where scientists around the world publish their sequence information. GenBank is not the only sequence database. There are other certainly important databases such as the Barcode of Life databases, especially when we're talking about DNA barcoding. Barcode of Life database is run by the University of Guelph outside of Ontario, Canada. And they actually focus really exclusively on DNA barcode sequences. Now, in the past, the sequence data that you found on the Barcode of Life database and the sequences that you found on GenBank were somewhat mutually exclusive from each other. But recently, I've seen that GenBank will start to import sequence data from BOLD, and BOLD will import sequence data from GenBank. So you don't have to do a lot of comparisons or checking of your DNA sequence across multiple databases now. Most of this data can be found in GenBank. Now, we used to be able to connect to, I should say we tried to connect to GenBank in real time, but you can imagine how large that database is. So that took quite some time for us to do an alignment through our servers here. So what we now do is we pull in a subset of samples from GenBank that's generally that's very similar to the types of samples that we search for. So again, we search for plant, we search for invertebrate. So we're not necessarily bringing in viral DNA in here, which is something that we wouldn't normally look for. So that narrows down our database. It's still looking at sequences available in GenBank, but it makes it a much faster sequence comparison because we're not directly connecting to GenBank in real time. Okay, so I'm going to go ahead and click on BLAST. And you'll see you have the option here for blasting the paired PWH002 sample, the paired RKH002, or the individual reads for PWH009 forward or reverse. Remember, forward was the low quality one. There's no reason to blast any low quality sequence data. It's the #1 thing that students really love to do. They're trying to get as much sequence data as possible, but we can have no confidence in that low quality data. So certainly make sure to tell students that if it's low quality, there's no reason to continue processing it or blasting it. Okay, so I'm going to go ahead. I'm going to click blast for my first sample here. Now, this might take a little bit. So sometimes it takes a few seconds, sometimes it takes 20 seconds, sometimes it might take up to a minute. If someone has run this data before, if they've looked for this sequence, in the database, it tends to remember that analysis that was done, so it tends to be a little bit faster. If it's a completely brand new sequence that it hasn't tried to align before, it might take a little bit longer. So just be aware, you might need to give it a little bit of time to do these blast analyses, a minute or two sometimes for each sample. Okay, so what are we looking at now? So what it's going to provide to us is the top 50 closest matches to that query sequence that we tried to compare to the available data on GenBank. So first here is the accession number. The accession number is basically just an ID that is assigned to any sequence data that is published to GenBank. This is how you can track or search for sequences specifically by an accession number. I'll come back to the details in just a moment. Alignment length, this means how much of your query sequence matched with an available sequence on GenBank. So the larger the alignment length, the more sequence that it was able to match to. Okay, so usually the alignment length I find with our DNA barcode sequences tends to be very, very close to the query length that we blasted. Mismatches are super important. So mismatches tell us how different our DNA sequence was compared to these closest matches. So of course, 0 mismatches means that we're identical sequences that we compared. And then as we see an increase in the number of mismatches, that means that we had some sort of DNA polymorphism in there. So it means a change in a nucleotide somewhere within that DNA barcode region, okay? Bit score and e-value, things I don't want you to get bogged down with, but I do want you to understand. So the e-value is the expected value. So this is really the probability that the sequences that were returned here in these top 50 matches are due simply to random chance. So again, like I was saying, that example ATTA, what's the probability that the first result here was in fact our sequence? These e-values are scored based on the size of the database that you're searching. And the lower the e-value you have, the better I should say it's considered to have little background. So you want a very, very low e-value. So what we're seeing here for my samples are 0, okay? So what that essentially means is that there's almost 0 probability that the results that we've obtained from these 50 closest matches are due simply to random chance or some sort of background. Okay, so very, very small numbers or 0 is what we're looking for the e-value. And I would say for the majority of your barcode samples, you're going to get something that's very, very small or 0 here. The bit score is also a measure of chance, but it looks at the sequence quality independent of the actual query length of your sequence and independent of the database size. So the higher the bit score you have, the more significant the matches are. So these are high bit scores. You'll see usually they'll be maybe, high 6, 7, 8, 900, or even, 4 digits long. I don't have a great cutoff for what's considered a low bit score. I was told once that as long as it's 3 digits, generally that's considered a good bit score. But I would say what we're seeing here is generally pretty typical of what a good bit score would be. Okay, so lastly are the details. So the details are the information and hopefully the identification of our species. So what we're seeing here is the species Tetramorium cispicium. Sorry, my Latin is terrible, so I'm terrible at pronouncing these names. So if anybody knows better, please do, in fact, tell me the actual name of these. So this particular Tetramorium species, you can see there's a number of these results that came back. There's a number of Tetramorium cispicium species that came back. And this is because there's no limit to the number of, or the same number of species in GenBank. So this could be different scientists from around the world uploading sequence data. It could be someone who's looking at sequence analysis across a broad number of samples that they collected and they published all of them to GenBank. So this is why you may end up with a bunch of the same species that you're seeing, all with 0 mismatches. So I would say we'll go ahead and scroll through here. And you can say for all the top 50 matches, and we start to get into a few polymorphisms here. So we'll see that there's others, it's the same species, but there's some obviously that have some polymorphic differences here. But I would say we could be pretty confident that we've in fact identified our species. We have 0 mismatches and all the closest matches to it, at least the top 50 here, are in fact the same species. So this would be a good, I'd say we were pretty good at identifying this particular species. If you hover over the name here, you can actually see it will give you the full taxonomy of your organism, which is nice. And if you click on it, it usually will pull up some additional information. So you can click on any of these links that will allow you to just take you to more information about that organism. And every so often too, it'll pop up here with a picture of the organism. It simply depends on what was uploaded for this particular organism that when we were searching the database. If you were looking for pictures and it doesn't pop up when you click on the details here, I do what pretty much everyone else does, and I simply Google it. So I'll copy this, I'll paste it into a Google search, and I'll do maybe a Google image search to confirm that this does in fact look like the species that I've collected. Okay, so that's sample PWH002. I'm gonna go ahead and blast, I'm gonna do the reverse here of PWH9 first. So I'm gonna, again, this is just a single read that we're blasting now, and you'll see that we have, again, we'll get the top 50 results that came back pretty quickly. You'll notice again, high bit score, low E value. We'll see a few different mismatches here. This will always align initially, or excuse me, I should say, this will show you the closest results by the alignment length. So it'll show you the alignment length first. What I generally like to do is sort by number of mismatches. So you can use these arrows here on either side and sort by the smallest number of mismatches. As long as the alignment length doesn't change very, very much, then you can still be pretty confident in the details that pop up here. So I can see this particular sequence was only one mismatch, one polymorphism away from the closest species here. And that makes sense, right? Because I didn't do any trimming on my sequence because I was only using one read. So if I were to be able to go back and trim off those ends, I bet I would get rid of these mismatches here. And in fact, it would actually be 0 mismatches for this particular species. But even if it weren't, even if it were 1 mismatch or 2 mismatch, it doesn't mean that that's not your species. In fact, generally it does. So if you just a couple polymorphisms here can still reflect the same species. Now, people generally ask, how many differences does it take before you know when it's a different species? And that's a really hard question to answer. Why is that? Because it depends on the taxa. So for example, a lot of plants tend to be highly conserved in the RBCL region, barcoding region that we use. So something as small as 1, 2, 3 polymorphisms could be indicative of a completely different species. Whereas with some invertebrates, you could have 20, 30, even 40 mismatches, and it still be the same species. There just happens to be a lot of polymorphisms and a lot of variability within the barcode region of that species. People use the rough cutoff of about 95% similarity. So if it's less than 95% similar, they'll consider that to be a novel sequence. So something that is unpublished to GenBank and potentially a different species than your closest match here. But just because it's 94%, or excuse me, I should say 96% similar, doesn't mean that it's not a different species. Or if it's only 92% similar, doesn't mean that it isn't the same species. So if you have any questions when you're analyzing your data about how similar, or whether or not you have a novel sequence, or if you think the closest match is in fact your species, please do reach out to us when you're doing the analysis, because this is why we have Jeff and Sharon. Because in addition to us doing the DNA barcode, they will also take a look at your actual samples and do the taxonomic analysis so that we can get a good idea comparing your DNA barcode region to their taxonomic analysis so that we can get the best identification of your ants as possible, okay? But I'd say here, if we scrolled through, we can see this Monomorium, again, emarginatum species. I'd say that was pretty much the first 20 or so, and then we're starting to get this Solenopsis. So that's a little bit different, but we're getting more mismatches here. So there's a good probability that this Monomorium emarginatum is in fact our species. But we can always, again, look at the pictures that are available for this, compare it to ours, and have it taxonomically identified to be sure. Okay, lastly, I'm going to go ahead and blast this RKH002 sample. And we'll see what happens. Okay, so now I'm going to go ahead and sort by number of mismatches. Now we see something very different. Okay, we see the largest number, or sorry, the smallest number of mismatches we have here is 57. That's really, really high. So what does this mean? Well, it means that the closest match to the species, to the sequence that we blasted, is this Polyergus breviceps. But is that in fact our species? Well, for this particular sample, we had a taxonomist identify these samples even before they sent them to us. And this individual, John Marlin from Illinois, who sent us these samples, actually identified these samples, this particular sample, as Polyergus montavagus. Montevages. And so obviously that's not the closest match that is here available in GenBank. So I went ahead and looked in GenBank to see if there was sequence data available on Polyergus montevagus. Is this just a really, does this have high polymorphisms compared to that one for some reason? In this case, no, there is actually no barcode published for Polyergus montevagus. So this is not only a novel sequence because it's very, very high in the number of mismatches, but it's a completely new GenBank record. So this was some of the first data that we got back from this US Ants project. So we're certainly excited to see what the rest of your projects are bringing in, because I think we're going to see a lot of cool stuff like this as well. So what we will do then is and for all of your sequences, we will do this, but especially we'll indicate whether or not the sequences are novel, whether they're novel GenBank entries, whether they're a polymorphism. So even if there's 1, 2, 3, 4, 5, a few differences in your DNA sequence, or even if it's identical, we will still publish all of your sequence information to GenBank, because even that will provide us GPS records. So for many of you, remember, we had these targeted hit lists where your sequence for your ant might be available in GenBank, but you may have collected it in a region where the distribution of that ant is not common. So providing that information to GenBank is also important, that GPS location information. And in fact, we've had people contact us before who have seen our sequences available on DNA Subway and have said to us, that's really interesting. We've never seen that particular species in the Long Island Sound. Can you provide us with more information about your sample so that it does not go unnoticed? So all of this will get published, but this here is an example of a novel sequence and in fact a novel GenBank record. Okay, any questions about that so far? So that's BLAST, that will be the identification aspect of your particular species. Is everybody with me? Mary, go ahead.
One quick question. Okay, so you said that you either sequence the forward or the reverse, not both. How do you know which one to pick? I'm sorry.
So for all of your sequences, we will sequence both the forward and the reverse. So hopefully what we're hoping for is that you will get that consensus. So when we did the consensus builder here, it will compare it, it'll give us the consensus. And then when we blast, it won't even give you the option to select forward or reverse.
Oh, okay. Okay.
Only time it gives you that option is if we can't build the consensus because one or both of those sequences are low quality.
Okay. And one more quick question. So you should never, obviously trimming is the best, especially when your sequence is long enough where you don't have to guess. But let's say it's kind of small, and do you ever recommend manually changing a base if you feel pretty confident from the waveform, or no?
Okay, so that's a great question. So I'm going to go back into Consensus Builder. Now, if for some reason, let's say that it chose this particular C here, okay, and it said that this was a C and that this was a C. But if I looked at that and I said, no, actually, I think that's, a T at that position, I do have the option to change it. So I can click here and click T and it'll highlight it in pink.
Okay.
I would say in the 5.5 years that I have been using DNA Subway, I have done that once.
Okay.
So generally, it either does a better job of choosing the consensus, or if it's at the either end, I just simply trim it out if I'm not sure. But yes, you do have that option. I don't generally recommend it because I think the software does a good job. But of course, it's a computer, we're human. If you feel that you have, that you can make the better determination, then yes, you do have that option. Good question. The other thing I want to mention that I didn't bring up before is if, say, you blast your sample, I'll just open one of these here, and you get a number of different species, all with 0 mismatches. What does that mean? Okay, how do you decide which of those species it is? Well, if they're all 0 mismatches, that simply means that you had a high degree of conservation between your barcode region and of all those species. Or alternatively, you were blasting a very short DNA sequence. So where you had those polymorphisms that could distinguish from one species to another, you were missing some of that sequence data that would normally help distinguish that. Okay, but I would say that usually it has to do with the level of conservation. So how might you tell between 2, 3, 4, 5 different species that all have 0 mismatches? Well, again, that's where Google image search is your friend. So you'll take a look at those high quality pictures that you took and posted to the sample database. So you can use that for comparison. And of course, like I said, we have Jeff and Sean here today who can also, in our project, I should say, who can also look at those samples and help to make a better taxonomic determination. So in that case, usually if you see that multiple species matches, I would say for the large majority of those samples, they usually have the same genus. So obviously there's just not a lot of differences between the different species within one genus. Can it be different genuses? Sure, general, sure. But usually I'd say that it's just a matter of different species between the same number of, or I should say the same DNA sequence, but the different species within one genus. So that's the other thing that you'll tend to see. Now, if you get what we saw here for RKH002, if you get these large number of mismatches, especially if you're working with students, the first thing have them do that you should do, go back and look at the sequence quality, okay? 50% of the time when I have students contact me and tell me that they have novel sequences, it's because they blasted low quality sequence data. So just confirm it. Then if it is high quality and you get here and you have the good bit score, you have the good e-value, and you still have the high number of mismatches, then that's a higher probability that you have a novel sequence. Okay. Okay, so what do we have left under add sequences? Well, we have this option here to upload data. This is just if you wanted to add additional sequence data to your project. So for example, if we provided you with a tracking number and maybe one or two of your samples was low quality and GeneWiz redid them, and you didn't want to have to create a brand new project and reanalyze all of your sequence data, you could simply come in here, click import trace files from the DNA Learning Center and add additional samples by just simply pasting your tracking number in here again. So it's an easy way of adding additional data. What else can you do? Okay, you can import sequences directly from GenBank. So if you wanted to compare to sequences that may be available. So for example, we've had organisms before where the closest match is not the species that we know it to be, but that species sequence data is available in GenBank. Okay, so we want to bring in maybe some of those species directly from GenBank and do an alignment, look at all of those sequences. You can actually do that by adding the accession number here. So remember when we did BLAST, go back here, that accession number is here. So you can simply copy and paste an accession number that you get from GenBank into that upload data and we'll pull in that data directly for you. You can do the same thing from the Barcode of Life database, which I mentioned before, using the process ID. It's basically just the Barcode of Life database's same term for an accession number, same thing. It's just an ID that's assigned to any sequence that's published in the Barcode of Life database. You can also upload again or enter those sequences in FASTA format. So again, that's the raw sequence data that's been given a title. So for example, it would look something like you would have a caret, you would have name of sequence, so whatever your sequence was, it would be a hard return, and then it would be, you know, your sequences, however many sequences that was. So that's what the FASTA sequence looks like, and I'll show you where you can get that in just a bit. Okay, so nothing, you may not, for the most part, need to use this upload data, but it's available to you if you want to add more sequence data. Okay, lastly, under this column is the reference data. So this is where you can bring in data that we have available to you on DNA Subway. And in fact, what we did is we added a US ants reference data analysis. So we picked ants from across the country that we could use for comparison or analysis with our particular sample. So you can bring this reference data in, and this you can use for when you're doing alignments. If you want to compare your sequences to other species, you can also use this for building phylogenetic trees if there's a good reason to bring this reference data in. So I'm going to just click on US ants, click on add reference data, and now it'll tell me that it's added to my project. Okay, you can bring in anything else, but again, doing alignments with some of the mammals, fish, sharks, you know, for ants is probably not going to give you good alignments or make for an interesting phylogenetic tree. You can look under common insects, that would be more reasonable, but we specifically added ants for this project. The other thing I want to show you back in BLAST is how to add sequence data from your BLAST results into your project. So I'm going to do it now just for let's see, we'll do it for PWH002. So I'm going to bring in a bunch of these sequences. And you'll notice next to the accession number here, there are these squares, these boxes. You can click on those and it'll ask you if you want to add these blast hits to your project. So if you want to do a sequence alignment and compare your sample to your blast hits here, you can bring in all of this sequence data. So this is really will make for, again, for nice alignments and nice phylogenetic trees. So I'm going to go ahead and just bring in a couple different or a few different ones here. You can bring in as many or as few as you want, depends on what you're trying to analyze. And then I'm going to bring in some of these ones that were a little, had a few of these polymorphisms in here, and we'll do a comparison. So I'm going to hit add blast hits to project, and I'm going to do the same thing just for the PKH002 here as well, because that one had a lot of those differences. So I'm going to bring in the closest, this Polyergus, which is still 57 mismatches. And then I'm also going to bring in some of these other ones that had differences in the numbers of base pairs. And again, for now, I'm just doing this kind of arbitrarily to demonstrate to you. But again, you would have more thought as to why you were bringing some of these blast hits into your project. Okay. So we're done here with adding sequences. The next is selecting sequence data. Okay, this is where we can do sequence alignment. So we can look at all of the sequences compared, our blast hits compared to our sequence that was our query. We can look at our reference data compared to our sequences. So the first thing I'm going to bring in is my PWH sample. I'm going to bring in the blast hits for that sample. That was all the ones that were that tetramorium. And then I think I'm just going to bring in a few of these US GANs. I don't want to bring in all of these because this is going to overwhelm my sequence alignment for my tree, okay? And then I'm going to save those selections. And here we have muscle. So muscle is an algorithm that's going to take all of my sequence data and try to align it to create the best fit so that we're looking at the same region across all of my sequences. So this is very similar to if you use like the clustal alignments before. It's doing this the same type of algorithm here. So I'm going to click on muscle. It's flashing, flashing, flashing. And then it's done. I'm going to click on it again. And what you see now is, ta-da, okay, we see our sequence alignment. So what is this? Well, first of all, this kind of looks like one of those kind of UPC codes, right? Like a barcode. So this is in part where DNA barcoding gets its name. What we're seeing is a pictorial representation of the differences in our DNA sequences. So anywhere you see a colored bar here, red, green, blue, or black, that's representative of an A, T, C, or G at that position that is different than than some of the other samples that you see here. So what you can see is that what this alignment does is it will group together the more similar organisms that we're seeing down here compared to the different ones. So why are these so different? Well, because this, my sample here, PWH002, which is right here, okay, was more closely related to these other blast hits that I brought in than they are to that random reference data that I brought in of the other ants. So obviously a high variability within all of these different ant species here, but we're seeing here very, very similar DNA sequences between my organism and its closest blast hits. If this is hard to visualize, you can toggle up front here between the barcode view and the base pair view. So if I scroll over, you can see now exactly the differences there are between all of the sequences. So for example, at this position 100 here, you can see that these other ants that I brought in from the reference data, they all have an A at this position, but my sample and all of its blast hits have a G instead. That's what it chose for the consensus sequence. So that's, all of these are A, all of these are G, and that's why it's grouping these together, because their sequence data, as you can see by the consensus sequence, is much more similar to each other than they are to the reference data. Okay, so that might be a little bit easier, especially again, if you're working with students, for them to understand sequence alignments and the differences between the DNA sequence. Okay, but barcode view allows us to visually and kind of rapidly look for the grouping of different organisms as we're seeing here. You also can look at sequence similarity, so you can compare the actual percentage, how similar are the organisms to each other. So again, I mentioned that kind of 95% cutoff that you might use to determine if you have a novel sequence. Again, that's up in the air depending upon the species or even the taxa that you're using. But sequence similarity, if I open this chart here, remember my sample PWH002 was most closely related to what was number 7 here when I did my blast. So if I scroll over for number 6, I'll just highlight this, or will it let me copy it? Let me see. Sometimes it lets me. What? No, it's not letting me today. So I'll just have to scroll over. So if I just go across this line here for number 6, and I match it up with number 7, okay, so this is 6 here on this axis and 7 here on this one, you can see it's 100% identical. Okay, so that's the same 100% identity. If I compare 6 to 7, 8, 9, 10, and then it gets a little bit different from 11 and 12. Why is that? Because 11 and 12 have a difference here in this nucleotide. So they were different. They're close, still closely related, but there was a polymorphism here. That was that one mismatch that we had. Okay. All right. So now make this smaller again. The other thing that we can do is build a phylogenetic tree with this. So we can trim the alignment. Okay, and what we have to trim the alignment before we can build a phylogenetic tree, because we have to ensure that we're looking at the same size data. Okay, so we're all looking at exactly the same region when we're building this phylogenetic tree. So that trims off all of those dark gray regions that were on either edge here. So now we're looking at exactly the same amount of DNA sequences between my sample, its blast hits, and the reference data. Okay, so I'm going to close out of alignment viewer here. And I have 2 options here to build my phylogenetic tree. I have PhyLip-NJ, this is the neighbor joining model, and I have PhyLip-ML, this stands for maximum likelihood. So PhyLip-NJ, the neighbor joining model, this is a phonetics type of building a phylogenetic tree. It is less computationally expensive than using the maximum likelihood, and it tends to it tends to build a tree based on similarities in characteristics or similarities in sequence of the organisms. Whereas the maximum likelihood model brings in evolutionary relationships. So it looks at the evolutionary characteristics of organisms or their DNA sequence. Okay, so neighbor joining is a quick, simple model. Maximum likelihood tends to do a better job of, I think, organization of your data and tells a better story. But we'll look at both. So if I click on the neighbor joining, we're going to build that first. We can open that. First thing that you have to do is select an outgroup. So an outgroup is essentially the root of your tree. We say that, like, if you were looking for an outgroup in your family, it might be like a great grandfather. It's an individual who is somewhat related to you, but more distantly than, say, an immediate sibling, child, parent. So your outgroup here, this is one of the reasons we brought in the reference data, was we can bring in another ant. Obviously, ants are, distantly related to each other at some point, but all organisms of the same genus are going to be much more closely related than organisms, other ants of a different genus. Okay, so I'm just going to select one of my, doesn't matter which one, I'm going to select one of my organisms as the outgroup. Always make sure that your sample or your blast hits aren't the outgroup, unless you don't bring in reference data and you just pick a blast hit that is very, very different from your sample. So for example, my one that was 57 mismatches, if I picked the one that was the most different in that group, which I think was like 85 mismatches or something similar, if I brought that one in, then maybe I could make that one the outgroup instead, and the other results then would fall out of my tree. Okay, but I selected one as an outgroup, and what are we seeing here in my tree? We have the outgroup will be labeled in red here, and I'm looking at my sample PWH002 compared to the closest blast hit here. Now, what you'll notice is that it forces what are called sister clades between these 2 organisms. So it's pairing these 2 branches to each other, even though we know that PWH002 is actually closely related to a number of these blast hits. It had identical sequence data to a number of these blast hits. And that's being represented here by these numbers. So these numbers are called bootstrap values. And essentially, when it was flashing and creating that tree, it was creating 100 different trees while it was flashing. And 16 out of 100 times, it paired our sample here with this blast hit, with this result here. So why wasn't it higher? Well, because some other times it paired this one with this one, okay? Or some other times it paired our sample here with this one, because they're all identical. So what did it do? It did a reasonable job of grouping these together. So you can see that all these ones are grouped, so they must be closely related. These other two that are grouped down here, those are probably the ones that had the single polymorphism, but it's not giving us any information about the evolutionary relationship between all of these samples, okay? So let's instead do the maximum likelihood model. Open that up, select my outgroup. I'm gonna use the same outgroup, so it uses the same base of my tree. And now what do we see? Now we're not forcing those sister clades, so we're not forcing it to branch, and we can see my sample is on the same branch as 1, 2, 3, 4 of those other blast hits. So this is showing that all 4 of these right here are identical to each other in their sequence. And then they're closely related to these other 2 that are down here, okay? So you can see that branch length here means changes over evolutionary time. So the branch length is actually meaningful here as opposed to in the neighbor joining model where it wasn't meaningful. So these are all closely related, and this very small difference here, what does this mean? That was that single polymorphism that distinguished these 2 samples here from these other ones here. Okay, so this is a much better representation of those data. But again, it's more computationally expensive. So if you have a lot of samples that you're trying to build a tree of, this can take a lot more time to run and to try to build a tree. So that's why people will sometimes use one model over another, depending upon what question they're trying to answer. The last thing that I want to do is just quickly, 2 things I want to show you real quick. I just want to show you, if I selected this sample here, instead of saving my selections, I also have the option to download. So if I download, can anybody see, can people see the Word document that just opened? I might have to share my screen instead. Let me share that document. Okay, so can everybody see the Word document now? Okay, so what I did is I downloaded the FASTA version of that consensus sequence here, okay? So again, it's the caret, it's my sample, PWH-002. This is that raw consensus sequence. So if for some reason I wanted to take this sequence data and I wanted to blast it directly on GenBank, or I wanted to take this sequence and look at it on a different database and look on the Barcode of Life database or another database, I can now just simply copy this sequence data and paste it anywhere that accepts FASTA sequence, okay? So this is what I meant when I was talking about FASTA sequence before. It's simply the consensus sequence with a caret, the name of the sequence, a hard return, and the raw sequence here, okay? So you do have that option of downloading the sequence data. The last thing, I just want to show you what the phylogenetic tree of the one that was very, very different. So I brought in the closest blast hits here. And I'm going to bring in a few of my reference data again. Okay, save those selections. I'm going to do a muscle alignment. Again, it's very different, so that's why it's taking some time now, because it's really trying hard to find a good consensus between all of them. And you can see, I'll trim it to make it a little bit easier to see. You can see here's my sample now, that RKH002 sample, and its closest GenBank hits are surrounding it. So there was those Polyergus breviceps samples, and you'll see that it's not very similar to any of them. Okay, so it's very, very different. And I pulled in the closest GenBank hits to that particular sample. So again, it's just a good pictorial representation that our organism here is very, very different. Okay, and if I were to do a tree, okay, we can now see here's my sample on a branch all by its lonesome. Okay, it's showing that it has some evolutionary relationship to those Polyergus samples, but again, it was a very different Polyergus sample. It wasn't the breviceps, it wasn't the samurai here, which is such a cool name for an ant, but it was that Polyergus montavagus that is unpublished. So it's got a relationship with these other organisms, which makes sense. It's the same genus, but it was a very different species. And both my sequence alignment and my phylogenetic tree is a good representation of that here. Okay. So I did pretty good for me. I only went 6 minutes over. I'm quite the talker, so sometimes it extends a little bit. But does anybody have any questions about how to analyze your sequence data? I just want to show you, when I say, I stopped talking, just kidding. we will provide for you a means to organize your data. So when you're going through your sequence analysis, we really recommend putting it in something like an Excel file. So here I have an example where we'll have your team name, your location, your sample IDs. If you've identified it yourself, if we had a professional identify it, was your PCR successful, was the sequencing successful, putting your tracking number in, and then in adding the information like what was the closest, blast hit or hits? Did you have multiple that were 0 mismatches? How many mismatches were they? What was the percent identity on those? What was the accession number of the closest match hit in case we want to bring those in and build trees? Is this a novel? Does it appear to be a novel GenBank record, or did it have polymorphisms even if you were able to identify it? So that will give you some sort of form similar to this so that you can organize your sequences in a place that makes sense. And then lastly, I have a couple of resources here. So we have a number of videos. I mentioned at the beginning, some walkthroughs, some training, some tutorials that are available to you. I will put them in the Google Classroom, of course, but they're also available on the website that I sent out in, I think, 2 emails ago, the DNAbarcoding101.org website, where we now have a citizen science section. So that is available. Videos, this is of course Dave, who has a very, very nice webinar that analyzes sequence data, so you're welcome to use that. This is an 8-minute video that walks you through DNA Subway kind of very quickly, but if you need just a quick refresher, that's a nice one to kind of take you through it quickly. If you're looking for something more in depth, I have a DNA on the right here. You can see a DNA barcoding. It says DNA sequencing and species identification part 3. So I did a 3-part series over the summer on DNA barcoding. So this takes you a little bit more in depth into more sequencing. If you loved the sequencing much, you want to learn more about sequencing, that and sequence analysis. And then if you really, really love DNA Subway and you love the barcoding bioinformatics, another staff member here at the DNA Learning Center, Jason Williams, put together a really nice 3-part series, so 1 hour each of these 3 parts, that walks you through a lot of detail of DNA Subway and explains in more depth things like e-value and bit score, and takes you really in depth into some more of those components behind DNA Subway. So I certainly encourage you to review all of these resources. We have a number of protocols and videos to assist with the bioinformatics in addition to this training. And we do have clinics coming up, so I encourage you to, if you would like to attend a clinic, please do register using that link that I sent out in the email so that we know people are planning on attending the clinics and what you would like assistance with. So if you have any general questions about DNA barcoding, or if you have project-specific questions. So you try to identify your ant and you have some questions because either it looks like it's novel, or you're not sure which it should be, or you want assistance with getting it taxonomically identified. Again, we're sending all of those for taxonomic identification, but if you want to know the status, we're certainly here to help with that as well. Okay, so now I promise that's the end. Any questions about anything that we reviewed? I realize that's a lot of data that we went through. So, take your time going through it, use the resources we have available, but the more you practice with it, the more you'll realize that, again, if you go kind of just in that step-by-step manner, it's really a nice, streamlined, intuitive way of analyzing your data.
Sharon?
Yes.
This is Brian Wainscott.
Hi, Brian.
Can you quickly remind us how we're going to know when our sequences are available so that we can begin the process?
So when we receive your sequence data, you will receive an email from us with the tracking number and with the information on how to basically create your project. So you'll get an automatic email from us that will let you know that information. You can start accessing your project right away then.
Okay, thank you.
You're welcome. Anybody else? Okay, well, thank you everybody for joining me today, and best of luck with your sequence analysis. And again, we have a number of clinics coming up. First one starts this Thursday, so feel free to register for those for any additional assistance. And we are super, super looking forward to seeing what your sequence data comes, what we get out of this project, because we're already seeing some pretty cool stuff. All right, well, thank you for joining me, everybody, and have a lovely afternoon, and we'll see you soon.